So it is my pleasure to introduce the PhD paper award. So we had a number of proposals, and we selected David Meyer, who is from the University of Cologne in Germany, and who worked on aging clocks and the accumulating variations. So he's produced a very interesting model and new concept about age determination based on stochasticity. And I think that the best, anyway, is that he is going to present his work. But it was original, it was substantial, it was new methods. So this is really meeting the criteria that we're looking for this award. So I'm very pleased to introduce David Meyer.
Okay, thank you. Thank you for the really nice introduction and for awarding me for part of my PhD work. It's a great pleasure to be here, and it's a fantastic conference. So I'm David, and I would like to present to you aging clocks based on accumulating stochastic variation. And to start off with, I would like to start with aging. And you know that everyone of us ages. This can really happen at different speeds. So some of us age slower or faster than others. And the question here is why? And to be able to understand why, we first need to be able to measure this difference in the biological age. So how to do so? And for this, biomarkers of aging are tremendously important. Most well known are probably the DNA methylation sites that are known to change with age. And here you might find some biomarkers of aging that are biomarkers that tend to go up with age and some that tend to go down with age. But every single one of these biomarkers is really not that informative. So over the past years or a decade, machine learning models have been employed to use many of these biomarkers to highly accurately predict the chronological and now subsequently also biological age, something that we term now aging clocks. Now, one question in the field that really puzzled us is if aging is driven by entropy and the stochastic changes that accumulate with age, why can it be predicted so precisely? So the predominant thought in the field is that aging is driven by damage accumulation, DNA damage, loss of regulation, and so on. So why can we predict it with such a high accuracy? And this is something we try to figure out in our publication that came out last year. And we ask a simple question: how much could a stochastic process alone explain, or if at all? Maybe the model is wrong. And for this, we started off really simple. Pointer's not working anymore. Okay. With a stochastic process simulation. So we start here with a ground state, so just the starting point. In this case, it's completely artificial data, just between 0 and 1. But you could start with anything: transcriptomic data, DNA methylation data, and so on. And what we do then is really simple. We just simulate a stochastic process, which can be visualized by, for example, just a Galton board, where you have normal distributed values in the end. And we use now these small random values that are centered around 0, and we just add them independently to the ground state. Doing this once, we define to be one time step. So the ground state would be age 0, for example, and then doing this once is age 1. Now, we can, of course, not only do this once from the same ground state, but let's say up to 100 times, of course, making the data noisier and noisier with time. And now we use this data as training data, as input for example, an elastic net regression model that tries to predict the simulated age. So how often we applied noise to the data. And then, of course, we need some independent test data that starts from the same ground state. Everything else is the same. We just add noise to it. And then we want to see whether the model learned anything. And maybe not too surprising, in the beginning, we did not see anything. The prediction was completely flat. Stochastic variation accumulation without limits does not allow any prediction. Of course, because noise, by definition, does not have a pattern, right? But then we thought this is not really how biology behaves. In biology, we usually have some kind of constraints, some limits. DNA methylation, for example, is constrained in bulk to be within 0 and 100%. And now, just by restraining our simulations to be in some predefined limits, we see that we can build highly accurate predictors of the simulated age, although everything we do is essentially just adding noise to the ground state. Now, this was all completely artificial data, just a proof of concept. And now we're wondering, what would actual age predictors do? So if we have now an age predictor like an epigenetic clock, how would it behave on data with accumulating stochastic variation? And here the concept is essentially the same. The only difference is that we start with a biological sample as a ground state, for example, a young human DNA methylation blood sample, and the rest is essentially the same as I presented before. We used now this simulated data starting from the biological data as input for epigenetic clocks and want to see the outcome. The ground state, of course, should be highly accurately predictable because it's a biological sample. But what would happen now with the data where we simulated noise? And here, to my surprise, it is highly linearly correlated. So here on the x-axis, I show you the simulated age, so how often we added noise to the biological sample. And on the y-axis is one example, the Horvath epigenetic clock, the probably most well-known one, and the first one that was really popular. And it's showing a really nice correlation. And this is really true for every clock that we tested, irrespective of whether they predict the chronological age, biological age, or mortality risk. They all essentially just capture how noisy and less well-regulated data gets. And this made us wonder, if this works so well, could we do even the reverse? So can we use a simulated data set and use it to build a predictor of age that is then applicable to actual biological data sets? So we did exactly the same as I showed you before. We start again with a biological sample, we add noise to it, we have the training data, we train an elastic dense regression model that tries to predict how often we added noise. And then in this case, we use actual biological data from humans to see whether the model learned anything. I'm skipping here a few details. There are several ways to simulate the noise, but the core idea is the same. And here again, to my surprise, we saw that this works remarkably well. So stochastic clocks can really predict the chronological age of biological samples. Here on the x-axis is the chronological age of human blood DNA methylation samples. Every dot here is a completely independent human blood sample. And on the y-axis is the prediction output of the stochastic clock, which is just trained on this single one biological sample, really showing that it seems to capture something important. Now, this is chronological age. What about biological age? That would be actually much more interesting. Is that measurable as well? And for this, we had a look at this really nice publication that came out 2 years ago from Steve Horvath, so one of the key drivers of the epigenetic clock field, where they used pan-mammalian data to build a clock that not only can predict the age of one mammalian species to high accuracy, but the relative age of a large variety of lifespans. This is a figure from their paper where they show you here in the circle the species from the shortest lived mammal they tested all the way around to the longest lived mammal, and they show you here in the lanes around the circle the high accuracy of the clock with cross-validation, showing that the relative age compared to median lifespan is really highly predictable. So now we wanted to see whether we can do the same just with stochastic variation. And we used here the sample from a young dolphin species. So again, just one single biological sample, did exactly the same. And we see that for most species, we can actually recapitulate this and see that for most of the species, we see a highly accurate prediction of the relative age from short to long-lived mammals. It also captures known lifespan interventions. So if you treat mouse or also humans with certain interventions, it can, of course, affect lifespan. And here we see that for 3 different lifespan interventions for mice, so the growth hormone receptor knockout mutants, for example, or calorie restriction, we do see exactly what we would expect, that these mice live longer, so they have a younger predicted age, whereas smoking in humans tends to decrease the lifespan. And we see also then an increased predicted age. So here I show you 4 different clocks for different tissues. The difference here is really just how we simulated noise and how we started with. But essentially, we see that it's in almost all cases significantly capturing exactly what it is supposed to capture. Although, again, here we started just with one single biological sample. And interestingly, they also... Next slide. Thank you. They also capture reprogram dynamics. So reprogramming is, of course, a hot topic in the field at the moment. So you can reprogram old cells with Yamanaka factors to, in the end, all the way to iPSCs. but it has been shown that over the course of reprogramming, so if you treat the cells over a longer time, they gradually decrease the predicted age until they then end up in the iPSC state. And we see exactly the same. So with the stochastic clocks, we can even capture this artificial reprogramming. So the removal of noise is also captured, something that is was thought to be impossible because this noise is, of course, thought to be entropic, and many people in the field think that entropic changes should not be something that is revertible, but we do see with the stochastic clocks that it is indeed something that we can capture. So to conclude, I hope I could show you that stochastic clocks can be trained with one single biological sample, which makes them easily usable for rare data sets or species. So we don't need tens of thousands of samples. We can capture the same essentially with just simulations. They, as we think, capture the underlying principle of aging, so the loss of regulation, the increase in noise and damage. And this also not changed my view, because initially many people in the field and myself also thought that the genes and pathways that are being captured by these clocks, be it in linear or nonlinear clocks, are important, maybe also causal drivers of aging. But what we now think is actually that those genes and pathways that are captured by the clock are those that are allowed to accumulate stochastic variation at a faster rate, and might be even those that are least relevant. Of course, there are adaptive changes and responsive changes, but it seems that this is not the main driver of these clocks. They correctly predict aging trajectories across mammals, no lifespan interventions, and reprogramming dynamics. And something that I didn't show here, but we now put out as preprints, is they also capture naturally evolved rejuvenation dynamics, and also we can predict which neuron types degenerate earlier than others with this clock type. If you're interested in this, please also come talk to me. By design, they are not cell type or inflammation confounded. So if you train, of course, a normal clock on, let's say, blood data, you have cell type composition changes with age, you have an increase of inflammation, and many of these clocks are confounded by this. So if you train on normal blood data, you will find an increased inflammation, so these clocks will learn to associate inflammation with an increased age. Now, if you have a COVID infection and you test yourself, you are suddenly 30 years older, and this is, of course, not measuring the biological age, so what we want to see, but just inflammation state. Still interesting, but not what we are interested in. So I think these stochastic clocks can first of all be used to to validate aging clock predictions, because we don't have these confounding problems. And also they can be used in cases where just no clock exists. So if we have a rare species, the rare data type, this might work as well. In the paper, we went into transcriptomic and DNA methylation clocks, but in theory, it should also work in other data. And indeed, we have some indication that it might even work for image-based data. And with this, I would like to thank Björn, who was my supervisor during my PhD time, and of course, all of you. I would be happy to take your questions. Thank you.
Thank you, David, and congratulations. So any question? Yes, one here.
Thank you. Very interesting talk. So basically, your clock is trained only on one biological sample and then on the simulated data, right?
Yes, so we have usually start with one single biological sample, and we add noise in different ways. What I showed you here is just normal distributed noise, but we checked also other ways.
Okay, because I'm wondering, like by design, your simulated data is generated by adding noise,
Yes.
accumulated according to the age. So it sounds like your prediction will, like, according to the data generation process, is associated to the chronological age itself, and then the chronological age is like a big driver of the biological age. So I wonder, like, because you show the association between your prediction and the biological age, like, to what extent this can really imply the causal effect?
It's of course difficult to really show causal effects, but what we do see, for example, with the mammalian, These mammals, they have really different chronological age spans. So a mouse has like 2 years, and the whale like more than 100. So it seems like it's not really only related to chronological age, because then these smaller lived animals, for example, they would predict it not correctly. So what we do really think is that it depends on the maintenance system. So if you have a better maintenance system, which longer lived animals have, you repair your damage better, you can maintain it better, so DNA methylation just stays more stable over time. But if you have less well maintenance or less well DNA repair, you accumulate these stochastic errors at a quicker rate, if that makes sense.
Thank you.
So we have time for a quick question and quick answer.
Yes,
Please.
here. Thank you for a very interesting talk. So I wonder whether you can further improve your stochastic clock by limiting the analysis to certain genomic regions, such as CPG islands, shores, or shelves, enhances or repeats.
That's a good point. So the results in the paper, they were not really optimized to get the best predictions. So there's definitely room for improvement. It was more meant to show, like, as proof of concept that this is possible. But I think if you would spend more time to actually optimize these, and these are good ideas, I think it could be really interesting to, further develop it. Yeah.
Okay, thank you very much again, David.
Mhm.